Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Sina Sekheche, but I'm not alone today. I am with my co-presenter, Shabnam Sukmandan. And this lesson is brought to you by the Gauteng Department of Education in collaboration with Saibono Discovery Center. So like we started last week with our genetics, we did part 1. We did quite a number of things that was a mouthful with the new terminologies that you learned. So we are continuing this week again with the part 2 of genetics, which will cover the dihybrid crosses and the blood groups. So with the dihybrid, remember last week we did the monohybrid crosses and the sex-linked, but now we are doing the dihybrid crosses. So you'll see what type of crosses are dihybrid and also the blood groups. So at the end of this lesson, you must be able to know those. But just to remind ourselves that of things that you need to know before you enter into this, just to remind yourself of what we did last week. Remember, we said there's a format in which you write a genetic cross, and that format you collect marks by writing it in this fashion. So what is it that you need to look out for? The first thing, you need to make sure that you write your P1, and on your P1 you write the phenotype and the genotype. Where did we say you're going to get your phenotype? This one is a free mark because you are going to get it from the question. So they will tell you what type of parents are you crossing. P1 stands for your parent generation. Then the genotype, meaning from the letters, then you will decide whether they are homozygous. You will be told actually whether they are homozygous, heterozygous. You need to write their genotype in terms of letters. So this one you will also, in some questions you will be getting questions that have letters. In some questions you have to come up with your own letters. Then you know for meiosis and fertilization, these genotypes, they must undergo Mendel's second law of segregation, where the gametes are going to be formed and segregated, meaning you are going to separate them and you write them here. Remember, we wrote them in circles last week. And then these are your gametes after meiosis. Remember, meiosis comes first because it is the process where we are forming gametes. Then you write your gametes separated into circles, or you can put commas, but it must show the spaces, must be big. Then you fertilize these gametes during the process of fertilization. Then you can use a Punnett square, or you can just use that cross method, remember. Then for writing meiosis and fertilization at the correct place, you are going to get 1 mark. And then this, after crossing here, what you're going to get in these blocks of the Punnett square are your genotypes of your F1, meaning your F1 are your offspring. Now this offspring generation, you must write their genotypes as you get them from these answers that will be in your Punnett square, and these genotypes will therefore tell you the phenotype of these offspring to tell us what is their physical appearance. And therefore for writing P1 and F1 also at the correct places, you get 1 mark. So you have 1 mark for free for writing P1 and F1, 1 mark for free for writing meiosis and fertilization at the correct place, and this 1 mark that you get from the question when it tells you what type of parents you should cross. So please make sure that you write those, even if you don't know how to cross, or even if you are going to get the genotypes of the parents incorrect, make sure you write the structure correctly, because then you are going to get 3 marks for free. You can even get more than that out of the 6 that you're supposed to get. Right. So remember, we spoke about alleles last week, and now we want to see how do you write these alleles, and we spoke about them in a heterozygous state or in a homozygous state. Remember, they said they're always in partners, but it will depend which one is dominant, which one is recessive. We also explored the terminology of dominant and recessive. So let us take a look at this. You must be ready now to write them from the statement so that you can be able to work it out in a genetic cross. So let us take a look at these examples that we have here that is going to form part of our baseline, what we need to have going forward. Now, a dihybrid cross, or in a dihybrid cross, an animal with long ears, you see they've already given you a letter there, long ears. The allele that is going to represent long ears is represented with the uppercase or a with uppercase E, letter E, and red fur with R was crossed with an animal with short ears. So short is the opposite of long. So short will therefore be small letter E, and then black fur, which is now the opposite, which are using in terms of color, of red. Now the red in this case, the black is now with small letters. What did we learn about small letters and big letters? We know that big letters are showing dominant, and then small letters are showing what? Recessive alleles. Recessive alleles. So now we know if you have long ears, it means you have the dominant allele for long ears. If you have red fur, you have dominant allele for fur color. If you have black fur, you have recessive allele for black color, and if you have short ears, you have recessive for allele for short ears. Now remember what we said, that alleles work in pairs. They are always in pairs because they are from the mother and the father. Now you need to check the situation, that is it in a heterozygous or in a homozygous state. And remember we said for the recessive allele to be shown or to be expressed in the phenotype, they must be recessive, both of them, because if there is one that is dominant over the other, in this situation there is going to be complete dominance, meaning the dominant is going to be more complete over the one that is recessive. So let us take a look at our question for now. We know long ears, big letter E, red fur, big letter R, and then short ears, small letter E, black fur, small letter R. Now, which one of the following could represent the genotype of the parents? So this parent, the one with long ears and red fur, let us start with the first one, with the first parent. This first parent, we are told that they have long ears. So long ears, you expect them to have big capital letter E's. So if you take a look at this first scenario, do you have big capital letter E's? Yes, you do. And then they say this parent has red fur. The second alleles must be red fur. Let us see. Do you see red fur? Yes. So for parent one, for option A, it is the parent is correct. But we don't have only one parent. We need to check the second parent. What did they say about the second parent? This is a skill that you must have now going forward to be able to analyze from the question and be able to write the alleles. Right. They say the second parent, meaning this was crossed with an animal with short ears. So for short ears, what type of alleles are you expecting to see the second parent having? Small letter E's. Do we have small letter E's here on the second parent? Yes, we do have small letter E's. And then black fur, you expect this parent to have small letter R's as well. These are the R's. So can we say parent number 2 fits our criteria in the question? Yes, so this parent is also correct, meaning our A is going to be the correct answer. So our A over here is our correct answer. Right. Then there is a lot of terminology that we said we are going to do. So this is the same terminology that we spoke about last week. It's a whole lot of terminology, even for the entire topic of genetics. Remember, we discussed dominant allele, we discussed complete dominance, what is a chromatin network, what is a chromosome. Today we are here, we want to focus on this one because we are going to be doing dihybrid crosses. You need to know what is a dihybrid cross. So a dihybrid cross can be regarded as a genetic cross involving two different characteristics. Example, you can look at the shape and the color of seeds. So these two things are two characteristics. Remember last week when we were looking at our work, we were only looking at one characteristic at a time. If it was shape, we would only look at the shape or the inheritance of a shape in a particular species or organisms. But with dihybrid crosses, you are looking at two. Di means two. And please check the spelling here. The di here is written with D-I, not D-Y. So it makes a huge difference how you write this dihybrid crosses, because then it would mean something different if you write it in any other way. So please check the spelling of dihybrid cross. It means di means two. You are tracing now, or it's a cross where you are working or involving two characteristics. An example is that of one of the shape and the color. I think now you understand all of these we've spoken about. So let us go deeper into what dihybrid crosses are. We've built our baseline. We now know how to write our alleles. Then we are ready to do the crossing, because this one is a bit complicated. It includes a lot of gametes with the alleles inside. So you want to track them to be able to trace which one is which, so you don't get lost. So a dihybrid cross is a cross where there are two traits crossed. Remember, traits are also characteristics. This is another terminology that we use to talk about characteristics. Now, traits are inherited independently. Remember Mendel's third law. Mendel's law says, it is the law of independent and assortment, says that the alleles of a gene for one characteristic segregate independently of the alleles of a gene of another characteristic. What does this mean? It does not mean if one parent is, let's use the examples of the peas like we have on the side here. If one parent pea was smooth and green, they are going to give the same characteristics in that order to their offspring. So the inheritance of whatever characteristics is independent. You may find that the offspring inherits only the green color, the allele for the green color from the mother. However, they will inherit the roughness of the edges from the parent of the other, from the other parent. So you see, it does not mean they all come together. They are segregated. So these characteristics segregate, so they are randomly or independently assorted. None of these are dependent on each other. So they are independently assorted so that they can be inherited independently as well. Now, the genes are on different chromosomes. Remember, you have different chromosomes, so you'll find that there are different genes on different chromosomes. We located the genes, on the chromosomes, then we also located the alleles. You saw how they are positioned on the locus. Then when you cross a heterozygous organism, now this is always a condition. When you cross a heterozygous organism, what did we say heterozygous was? It is a condition where the alleles are not the same. There is one that is dominant and the one that is recessive. Because they are not the same, these alleles, for one characteristic, we say they are in a heterozygous state. So they say, if you cross a heterozygous organism with another heterozygous organism for both traits, meaning if for both traits it has to be maybe tall and round, that is the shape. Let's say round. You see, this big R and small r are heterozygous for the shape. This big T and small t are heterozygous for the height. So the two characteristics that we can check here are the height and the shape. So if they are in a heterozygous state like this, you cross them, you will always get the following phenotypic ratio in the offspring. Your offsprings are going to appear in this ratio always. 9:3:3:1. In always. Right. So I want us to look at an example. The only way to understand how dihybrid crosses work, or the genetic crosses, is to actually do them. Because when you do a genetic cross, that's when you get to understand all the terminology, all the concepts that we studied before. Now you are putting them into practice. You learn how to identify your alleles and how to segregate the gametes and how to cross them and how to get the genotypes and the phenotypes of the offspring. So let us look. So the key thing is for you to be able to analyze your statement or the question. In plants, tall with capital letter T and yellow flowers are dominant. So this information, they don't give you this information for free. You are going to use it. So you are talking about two characteristics here. What is the first characteristic? Is height. Let me write it in full, because if I write H there, you will be confused to say it's another letter that we are using. So this is height, and then yellow flowers. So this is the color of flowers. So there are two characteristics. Tall is represented with the capital letter T, and we are told that it is dominant. Yellow is represented by capital letter Y, and we are told that it's also dominant. They are dominant over what? Over short and orange flowers. Now here, you see you are not giving letters for short, and you are not giving letters for orange. Already automatically, because you were given the letters there to say tall is represented with the capital letter T, how is short going to be represented if it is recessive? Remember, if something is dominant over the other, it means the other one that is at the bottom is recessive. Meaning we learn here that short is recessive. What letter are we going to use to represent short? It is small letter T, because we're already given T for tall. And here, you do not use the letter of the word itself, like short. Some of you might be tempted to say the allele for short that is recessive is S. We don't work like that. The letter that you started with as a dominant one, you must write it in uppercase as the dominant one, and the opposite of the dominant will be the recessive, and the opposite of the capital is what? Is small letter of that same letter that you have identified in your answer. So small letter T in this case is going to be short. So that is also another skill that you need to have using your letters. Then orange flowers, we are not given a letter for orange flowers. What letter are we going to use there? We were told that yellow is capital letter Y is dominant. Then orange is recessive. Then we are going to use what? Small letter Y. We don't use a letter O, because then you will have so many letters that are confusing you. You will not know which one is heterozygous, which one is homozygous. It will confuse you. You work with the same letter for one trait. Yes, work with the same letter for one trait, one allele. Then that allele, whether you use the uppercase or lowercase, will be determined on the dominance and the recessive state of that particular allele. So now our flowers, we know they are tall and yellow, and there are others that are short and orange. A plant which is heterozygous for both traits was crossed with a plant that is heterozygous tall plant with orange flowers. You sit and you try to take it in. Heterozygous for both traits. What does that mean? Trait is a characteristic. So heterozygous for tall is going to be what? Capital letter T with small letter t. This is our tall plant that is yellow. It is yellow, a big letter Y and small letter y. So this is our first plant, because they said it is heterozygous for both traits. Trait number 1, trait number 2. Characteristic number 1, the height. Characteristic number 2, the shape. The color of the flower. Then they say it was crossed with heterozygous tall plant. Heterozygous tall. What does that mean? It means the alleles for height are not the same. This is heterozygous tall. With orange flowers. Orange. Orange, we said we represent orange with small letter y. And what you remember about the recessive phenotype is that for it to be shown in the offspring, it must be in a homozygous state, meaning our orange flower is going to be small letter y's, 2 small letter y's. Why 2 small letter y's? Because it is in a homozygous state, because orange is recessive. If you are going to write it with big letter Y and small letter y, what is going to be the color of our flower there? The flower will no longer be orange, but it will be yellow. Why will it be yellow? Because we know yellow, Y, big letter Y is dominant over small letter y. Once you have this as your genotypes, you will never go wrong. Right, so let us see. What do you do first when you try to solve a dihybrid cross? Step number 1, you might identify the phenotypes of the two organisms for each of the two characteristics. Now, phenotype is given in the question. They said what? So your P1 is going to be tall yellow flowers. They said heterozygous for both, but heterozygous means what? They are tall, they are yellow, and with what? Tall orange flowers. They are tall, but they are orange. So these are our parents. So you need to be able to identify the phenotypes. How are they looking on the outside first? Then you look for the letters that represent each trait in the question. Remember I said in some questions the letters will not be given. You will have to come up with your own letters, but it's very rare where you find questions like that. Now they will give you a letter or say using the letter T. Then you do the cross. So you must go to the question and identify the letters that are going to represent each trait. So for height, which letter are we going to use to identify the trait of the height? We are going to use letter T. This is for height. Then for the color of the flower, that is the second trait, we are going to use which letter? Y. Y for yellow dominant, orange recessive. So we have identified our letters. Then after identifying the letters, the third thing that you must do is to write the genotypes. So you are still writing the genotypes of your P1. So this is still your P1. How are they genotypical? You are going to get this information also from the question or from the stem. If they said parent 1 is heterozygous, it gives you a clue of how you're going to write your alleles here. Are you going to write them in capital letters? Are you going to write them in capital letter and small letter? Or are you going to write them in both capital letters or with small letters? You must listen or highlight and underline these terminologies on the question paper or on the question. When it says heterozygous, then you know not the same. Homozygous, they are the same. Dominant, recessive, those are the key things that will give you the correct types of genotypes. So in our case with these flowers, they said it is heterozygous for both characteristics. This is our parent number 1, remember. They said they are heterozygous for both characteristics, meaning what? The alleles are not the same for both characteristics. You see, there's more big letter T, small letter T, big letter Y, small letter Y. Then you cross them with a parent that is heterozygous for tallness. You see, this parent is tall, but does not have the same alleles that are homozygous. They are heterozygous. Then there's the orange. The orange is represented with the small y's. Once you have these now, what are you going to do? Step number 4. After you've determined the genotypes now, you must do your allele now independent sort. Different. You must determine the possible gametes that each parent can produce. Remember, possible gametes is during what is this independent random arrangement of chromosomes. So they are now falling in each gamete here, so you cannot have the same. So what are the possibilities of the gametes or of these alleles falling into each gamete? So we will work out all the possibilities of the gametes of the first parent and of the second parent. How do you do that? You do a cross multiplications of your first characteristic allele with your first with your second characteristic allele. So you are going to get what? Big letter T with big letter Y. So this is the possible first gamete. What is your possible second gamete? You take now the first T with the last Y. So capital letter T with small y. This is your second possible gamete. And then you come now to the second one, this T with this Y. You get this possibility of getting a small t with a big Y. Another gamete can have this type of situation. or you can have the last gamete with a small letter T and a small y. So all of these are your possible gametes for your first parent. So you are trying to do what? Random arrangement of chromosomes and segregation here. So you come again, you do the same with this parent, this T with this Y. This is the first possibility. And then this first T with second Y, another one. And then you have small t with small y, small t, then you have it like this. So all of these are now what? All of this that you have worked out here. Remember that you have not started with the genetic cross. You are now trying to do what? To determine the possible gametes. With these gametes that you have determined, you are still going to do what? To do fertilization. This is where now you put them in a Punnett square. And a Punnett square for a dihybrid cross is not going to consist of 16 or not consist of 9 blocks. It's going to have 25 blocks, meaning you are going to have a big Punnett square where you put all of these together, you cross them. Remember, gametes, fertilization, this is where now the gametes are fertilizing one another. So you're going to have all the chances of your offspring in those blocks of a Punnett square. Right. Step number 5, enter the possible gametes at the top side of the Punnett square. So for your second parent, you can put them at the top side. You can put maybe first parent. And then here you can put your second parent. It doesn't matter, vice versa, you are still going to get the same product when you cross them. So you will just use the first parent here, the second parent here. You put those possible gametes here. The ones that you've just worked out, you put them here at the top of each block. Then you also write the blocks going downwards. You put all those possible gametes. And then inside the blocks, you know, this is where now you are doing what? Possibilities of this gamete fertilizing that possible gamete of the second parent. What will be the possibility of the offspring, or what will be the possibility of the genotype in the offspring? That is what you get inside these blocks here. Right. So because of random fertilization, remember we said fertilization is random. That is why you're not only going to get one row. You're going to work out all the possibilities, because it is random. Gametes from both parents could fuse in different combinations to form the offspring. Hence, you have to have all your combinations of all the possible gametes from the one parent and the possible gametes of the other parent. It is because of what? Random fertilization. Fertilization is not just precise one. Now, in a Punnett square, you're going to write down the genotypes of the offspring that will result from each possible combination of the gamete. So whatever that you get here, these are the genotypes or the possible genotypes of the offspring from all the possible crosses that you are going to do. Step number 7, after getting the genotypes of the offspring, you are going to determine the phenotypes of the offspring. Where do you get the phenotypes? The genotypes obtained in the Punnett square are going to give you what the phenotypes are going to be like. So in our genetic cross, where we were crossing that tall plant that is yellow with a tall plant that has orange flowers, we get 6 that are tall and yellow, 6 that are tall and orange, 2 that are short and yellow, 2 that are short and orange. This is now the phenotype as you count from the genotype that you got. So I think it will make sense once we do it in practical now. Let us look at this example now of a dihybrid cross. A guinea pig with black long fur is crossed with a guinea pig with brown fur. Now you start to check, okay, black is represented with capital letter B. Already, automatically that tells you what? It is black is dominant with long fur. Long fur is represented with capital letter R, meaning long fur is dominant. Therefore, this guinea pig is crossed with a pig which is brown. See, brown here is represented with a small letter B. That will tell you that this is recessive. Then short fur, the opposite, small letter L. This is also recessive. Now, black long fur. Brown, short fur. Both parents are homozygous. The F1 offspring were interbred. Now use a diagrammatic cross to show the genotypes and the phenotype of the F2 generation, and as well as the phenotypic ratio. This is how a question can come about. Now, when they say both parents are homozygous, what does that mean? It means that for black fur, you're going to have BB, long fur, LL, you cross it with what? Brown, small letter B, with short fur, small letter L. Right, like this. So when you do your cross with these parents, you are going to get the first offspring. Now, after getting the first offspring, you are going to interbreed those first offspring, meaning they are going to act as parents. You are crossing the product. Remember, you cannot have this because you want the product of this. This were the initial P1. But from this P1, you need to determine what? You need to determine our F1 first. And then our F1 is what we are going to work with to give us the phenotypic ratio and the genotypic ratio of the F2 generation. Let us see it now. How do we do that? Right? So, you see here we have P2. What is P2? We don't have P1 like we always had because we took the product of the first cross. What was the first cross? Let's go back. The first cross, I just want to go back a bit. Okay. Right. Remember, our first cross was our parent from those that were homozygous, and they gave us the ones that were heterozygous, or what we will see when we go back, when we go into this question. Now, our P2, the products of our P1 are now acting as parents. Hence, we write this 2 here, not 1. So, how we are told that they were black with long fur, and then black with long fur. So all of them were black with long fur. Now, how was the genotype? The genotype now became heterozygous. Why is it heterozygous for all? Because they were homozygous. But remember, when you did the crossing with the first cross, you would be getting these as heterozygous now. So we work from this one, the one that we have right now, as a product. This is not from the question. You are supposed to work it out from the question first, and then you come to this and work it to be the second parents. So now the genotype, we are told that, okay, for black, you see it is heterozygous for black. Long fur, it is also heterozygous. Big letter L, small letter L. The same here, black, which is heterozygous, long fur, heterozygous. So our parents are like that. What do we do with these parents? We are going to find our gametes. The process of meiosis is going to happen to give us gametes. What are the gametes? The possible gametes that can arise. Unlike with the monohybrid crosses, the gametes you will just separate them here. But with dihybrid crosses, you must work out all the possibilities. Hence, you need to do a cross like this. You see, the first one with the first one here, this is where, let us take a look at them. So this first B with the first L. First B, first L. They give you BL. First B with the second L. They give you BL with a small letter L. Second B with the first L gives you B with big letter L. Second B with the second L, it gives you small letter B with small letter L. So here you need to pay attention to what you are doing, because what you are essentially doing is to multiply this one with this one, the first with the first, and then the first with the second, then the second with the first, and then the second with the second. You see, this is now all the possible gametes that you will get for parent 1. You do the same for parent 2. Here you got this one, big letter L, and this one together. B, L, and B with the second L, this one and this one together. Then second B with big letter L, this one and this one together. Then the second letter B with the second letter L, this one and this one. This is not your genetic cross. This is just possible gametes that you are going to cross. Then you will therefore then put them in a cross, a Punnett square. This is where you are doing fertilization to find out all the possibilities. So let us check all our possibilities. So when you take all of these, let's say all of this, and then you put them here, see? And then you take the second one, let's say this is our first parent, second parent, then you say this is our first parent, then this is our second parent, all the gametes. Then you cross them, if you cross this one and this one, you do the same thing that you do with the monohybrid crosses. However, here, remember, you have 2 characteristics. It is going to give you this big letter B with this big letter B, they give you 2 big letter B's. Big L with this L gives you what? 2 big letter L's. So I think you understand what is happening, how you do the crossing here. And the key thing is to write them, when you are writing them in a dihybrid cross like this, where there are 2 characteristics, the letters that are the same must be close to one another. What do we mean by that? If you have B, let's say, and you can't write it like this, BL, BL. Rather write it like this, put these 2 together so we can see they're in a homozygous or heterozygous state. So you can write them like this, small letter B's together and big letter L's together. So you must put the letters of the same alleles together. So even if when you cross you get it juggled or toggled, you will put them together so you can realize which one is homozygous, which one is heterozygous. Right, so when you do all these crosses, so these are all the possibilities of our offspring, the genotypes after fertilization that is random. What you do when you are crossing this, you are actually doing random fertilization. You are working out all the possibilities in this cross that our offspring will have. So let us take a look at the genotypes that we get. Remember, how do you get the genotype? When you look at this, the genotype here, it will tell you that this is black with long fur, black with long fur, black with long fur. How do you know? Because you know that dominant is long, dominant is black, recessive is brown, recessive is short. This one will be black with short fur. This one will be black with short fur, black, long fur, black, long fur. Then the situation differs in the homozygous or heterozygous. So you must be able to tell those. So let us check at the ratio. When we count all the blacks with the red furs and what? Long furs, we find that there are 9 that have black long fur, 3 with black short fur. Remember, you get this from the genotypes. 3 with brown long fur, then 1 with brown short fur. How were our parents? Our parents were heterozygous for both traits. And what did we say? What was the rule there? We said if you cross a heterozygous parents, what do you get? The ratio, you are always going to get it in the form of 9:3:3:1. And you see that here in this example. So you must keep that in mind. So when you do a Punnett square and then you find that no, the ratio for your offspring, when you cross the heterozygous parents, you are not getting 9:3:3:1. It means something went wrong. You must go back and check your genetic cross and try to fix it. Then when you look at the phenotypes, then you can say there are black long, black short, brown long, and brown short. So we have those types of offspring. So there are numbers in this way: 9:3:3:1. Remember, if you get big numbers, however, you're not going to get big numbers. For a ratio, you need to simplify it to the simplest. So this is another activity that we can do to check how do you analyze a dihybrid cross question. Right. In humans, short fingers, short in this case, with big letter F. C says short, but they write big letter F. What does that tell you? It is dominant. And the widow's peak, big letter H. The widow's peak is this thing that you have here. Are dominant. You see, you were able to determine that even before you went on with the question, just by the letters. You go back again, you check. Short fingers, our first characteristic. Widow's peak, our second characteristic. Both of them are dominant. What letters are used to represent them? F for finger length, H for the shape of the hair here in front. Those are the two characteristics. Over long fingers. What does it mean when they say over? Over long fingers meaning the long fingers are recessive. So, recessive. Then because they use big letter F, for the long fingers, what type of letter are you going to use? You are going to use a small letter F. And continuous hairline. Continuous hairline is that one that goes straight like that. Continuous hairline, the widow's peak is dominant over continuous hairline. And they use letter H to represent the dominant one, meaning the recessive one for continuous hairline is going to be small letter H. So you've just written those letters just by analyzing the first part of the question. Then you come. A man and a woman, both heterozygous. I said it is important for you to highlight words like this. Both heterozygous for the two characteristics. Now it's a walk in the park for you. When they say heterozygous for the two characteristics, you know heterozygous means the alleles are not the same. So for the first characteristics, that is short fingers or the finger length. So it must be how? It must have a big letter and a small letter, meaning you must have a big F and a small f. For widow's peak, you must have a big H and a small h. That is what is meant by both heterozygous for the two characteristics. Now, they plan on having a child. The table below shows the possible genotypes of the offspring. So they have already written the gametes for us, for these parents. So these are the gametes. They are crossing the gametes. You cross this one with this one. The possibility of having an offspring when you cross this gamete with this one is getting big letter F, big letter F. So when they have big letters, how is the phenotype? This is short fingers with widow's peak. So this first offspring is going to have short fingers with widow's peak. So that is the first one. The second one, if we go across, is going to have short fingers with widow's peak as well, because it is now in a heterozygous state and we know one is dominant over the other. The dominant will be the one that is expressed in the phenotype, right? Then the third one is going to be short fingers with widow's peak. Short fingers, widow's peak. So you can count from doing that. Short fingers, widow's peak. Let us take a look at this one. This one, long fingers with continuous hair. Why are we saying that? Because they are in a homozygous state and they are recessive. So you know that recessive ones, we said they are small letters. For small letter F, we know it's long fingers. For small letter H, we know it's continuous hairline. Therefore, by analyzing that, you can be able to tell how the offspring is going to look like on the outside. Now, state the genotype at Z. Here is our Z. Z, you are crossing this small FH with this small, this one, F, H, and this one. So at Z here, how is the offspring going to be like? It's going to have small letter F, big letter H, and small letter H. So that is the genotype at Z. Why? How is the phenotype there? You can say this is long fingers with widow's peak. You see, you are able to tell just by looking at that. Before you even go to the questions, learn to analyze what is happening in the information that you are given. Don't just go straight to answer the questions. Do the rest of the questions on this one, then we can just check. You see, we've put there at Z. We were correct to put those at Z. The genotypes of the parents. So how were the parents looking like? Remember, they said what? Both heterozygous for the two characteristics. These are our parents. So our parents, heterozygous for short fingers, heterozygous for widow's peak. Hence, we were able to get these types of offspring that we got here. The number of genotypes that could result in offspring with short fingers and a continuous hairline. Short fingers, what must you do? You must look for the ones that have big Fs with small H's and a continuous hairline. So you must count them. Big F with small H's. So when you count them from this Punnett square, you are going to get that it's 3 that are going to be like that. Then the allele for a continuous hairline. Continuous hairline, it is recessive. So what allele are we going to use to write for that? We did say small letter H. We identified this in the stem of the question before we even went to the questions. So you see it's why it's important to look at the questions, understand the question and analyze the stem before you even attempt to answer the questions, because therefore it will be easy for you to just write that information. You've established long ago that continuous hairline will be recessive, therefore recessive will be small letter H. The phenotype of a child who is homozygous recessive for both characteristics. Homozygous recessive, all small letters for finger length and for hair shape. So if you write all small letters, how is that child going to look like? The child is going to have long fingers and continuous hairline because they are all recessive. Long fingers and continuous hairline. I hope you were able to answer that one. Here is another activity that I would like for you to tackle as you work through your worksheet. Remember, you have the worksheet for today's presentation. Go through this activity, analyze your parents, analyze the types of alleles they have, homozygous, heterozygous, which one is recessive, which one is dominant, and then answer these questions so that you can test the skill that we've just learned in the previous activity. So please go do this activity. I don't want you to look through the answers. Here is another one as well. Also on this one, it is easier when you are given the letters. So the letters will guide you in order for you to be able to determine which one is dominant, which one is recessive, and then you can read how the parents were to give you this, and then you answer the questions. Mostly for the dihybrid crosses, you are going to get questions like this. In the examination, it might take time for you to write a whole Punnett square of 25 blocks and start counting. So they give you sort of like a short version of this, but you must still be able to understand how dihybrid crosses work so that you'll be able to answer the questions. The key is knowing dominant, recessive, knowing how to write your alleles in a homozygous or heterozygous state. If you get that, you master that, I'm telling you, you will never go wrong with these activities. Please go through your worksheet and do these activities. I'm going to leave it here now for my part of the session, but my fellow presenter is going to take you through the sex-linked and also the blood groups to check how do you cross the blood groups when you are doing genetic crosses. Which alleles can you use for the blood groups? Yes, then I will leave it to her now to take you through, but let us take a short break and then we'll continue with that after a few seconds.
grade 12 learners, welcome back to the second half of the genetics lesson. And today we're going to be doing blood groups and some sex-linked diseases. So let us look at some of the things that you need to know at the end of this lesson. You must be able to know the differences between an allele and a gene. You remember, you need to know that an allele is represented by an alphabet and that a height is, or a gene is represented by words. For example, gene for height, right? Height is a gene and it can be represented by a capital T or a small t. Depends, if it's dominant, it'll be capital. If it's small, it's recessive. So remember that alleles are forms of a gene or factors of a gene, right? They are factors of a gene and they are always represented by a capital or a small letter. Then you need to also know what is the genotype. You remember that is the genetic makeup, which you use your alleles to represent your genetic makeup. Alleles are used to represent your genotype, which is your genetic makeup. And you know your phenotypes is what you use to describe that organism, right? A tall plant, a short man, a man with blue eyes. So you use words to describe phenotypes and you use alphabets to describe your genotypes. And you remember you've just done the dihybrid cross, whereby you see two alleles will represent one gene, like big B standing for blue eyes and big R standing for red flower, right? Like that. Or that's how you actually look at the alleles and the genes. And you know there's two alleles in a dihybrid cross. Then you know we're going to look at now the different alleles for the blood group. So remember, as the previous presenter indicated, there's always two alleles. That means for one gene, there's two alleles. Why? Because when you talk about the height of a person, you are tall, but remember, you can either be tall or short, but if you are tall, the dominant one is what you see in the phenotype, but you may have your mother's allele, which is hidden in you. So you get one allele from the mother, one from the father. That's why that gene always has two alleles, a pair, one from the mother, one from the father. So we're going to look at the different blood groups, and we're going to use genetic crosses to determine the blood groups. Then we're going to also look at what are the sex-linked diseases that you've done last week, and that was hemophilia and color blindness. Right, now let us just look at some of the things that you needed to know from last week. You needed to know that some of the characteristics, right, that are displayed in a population, they could be recessive or they could be because of a disease like albinism or color blindness or hemophilia. These are not normal phenotypes of people. So they are caused by some kind of a gene mutation. So we're going to see that these gene mutations occur on an X chromosome. Right, now you're going to look at the sex chromosomes. You know, learners, that in a karyotype chart, right, in your karyotype chart, you always have chromosomes in pairs, and in the last pair, you're going to have your gonosomes or sex chromosomes, of which one must be an X chromosome. So your last pair is called your sex chromosome. In a female, it is XX, and in a male, it is XY. And you know that a sex-linked disease is when there's a gene mutation on the X chromosome of the 23rd pair. Again, a gene mutation on the X chromosome of the 23rd pair. And what do we call the 23rd pair? Gonosomes. And that gene mutation always occurs on an X chromosome, not a Y, because the Y chromosome is too small to have mutations and to have alleles and genes. That's why the mutation occurs on an X chromosome, and if the mutation occurs on an X chromosome, it's going to cause a disorder. So because the disorder is on the X chromosome of the sex chromosome, it's going to be referred to as a sex-linked disorder or an X-linked disorder. Right, genes located on the X chromosome. Now a father, can you remember that a father is XY? He has the last pair as XY. So remember, every individual or every offspring gets one allele from the mother that comes from the ovum and one allele from the father that comes from the sperm cell, right? So that, let's say for example, for height. So one comes from the mother, one comes from the father. Now, why the father cannot pass his Y chromosome to the daughter, because then that will become a son. So only sons and son-in-laws and fathers and father-in-laws and grandfathers have a Y chromosome. The males have a Y chromosome. So the Y chromosome can only come from the father, not from the mother. So if one parent donates one allele and if it's a son, that means the father donates the Y chromosome to the son. That means the X chromosome is coming from the mother. So if the mother has a disorder, then the mother will pass her genetic disorder to her son. Because the son will get a Y, it's normal. But if she has a disorder, and if that disorder is going to be showing in her phenotype, then she can pass the disorder to her son. So remember, sons get their sex-linked disorders from the mother if the mother has the disorder, right? And daughters will get the disorder from the father because the father can only donate his X chromosome to the daughter. He cannot donate a Y chromosome to the daughter. Just remember that, learners. So fathers can pass the X chromosome to daughters and the Y to sons. And mothers can pass the X chromosome to a son or a daughter because she got two X chromosomes. So mothers can pass the sex-linked alleles that form to both sons and daughters. That is the X to the power H and X to the power H. Let's say X to the power H is an allele. So X to the power H, capital H, is an allele. And it means she's normal. So this mother is absolutely normal. She's got no recessive alleles. So if she passes this to her daughter, she can also pass it to her son. They're going to carry the dominant allele of being normal. But if the mother is like this, she's XX, she's a female, and she's got hemophilia, and hemophilia is represented by a recessive allele because hemophilia and color blindness are recessive disorders. So they're going to be represented by two X chromosomes showing X to the power H, X to the power H, X to the power small h, X to the power small h. This X to the power small h is an allele. We don't just say H. That is a normal monohybrid cross. But when we say X to the power small h, X to the power big H, we are saying that this genetic cross is sex-linked. It's about a disorder. It's sex-linked. It's either normal or having the disorder, right? So in this case, this mother here, this one, she is a hemophilic mother. If X to the power small h means hemophilia and X to the capital H means normal, that means that this mother has two recessive alleles. She's homozygous for that trait. Two. Homo means same. That means she's carrying she's completely hemophilic. There's no normal allele to mask her. She's showing two recessive alleles and both are hemophilic. So she's going to be a hemophilic person in her phenotype, in her blood. She's not going to have fibrinogen to clot the blood. Remember what's hemophilia? It's a blood clotting disorder, né? Okay, so now, learners, you can see the mother can donate that X to the son and she can also donate an X to the daughter. All right, so that means the son can be hemophilic because if he gets a Y from the father, he's going to be hemophilic because this is the X chromosome and it's showing hemophilia. So he's going to be hemophilic. But if the son, that's XY, gets this allele, X to the capital H, from the mother, then he's going to be normal. All right, so remember here you can see that the son is XY and daughters are always XX, right? Hemophilia and color blindness are what? The two sex-linked disorders that you know about in our syllabus. There may be other sex-linked disorders, but that's not in the syllabus, but that does not stop the examiner from putting other sex-linked diseases inside your exam paper. As long as you must understand that a sex-linked disease is represented by an X chromosome to the power dominant allele or X chromosome to the power recessive allele. So they will indicate that in the paper. You don't have to worry about it. They will indicate nicely that this is the disorder and it's caused by a dominant allele. How you will know it is if they put an X to the power something, you know it's sex-linked. And if it's X to the power big H, you know that that disease is caused by a dominant allele that's causing that sex-linked disorder. So hemophilia and color blindness are always caused by a Recessive allele, so it's always going to be X to the power small letter. They can use any alphabet. They can use X to the power small d, small b, small h, small c, okay, small t. It doesn't matter, but that will be represented in your question. So you don't have to worry about that. So hemophilia is what? It's a sex-linked condition where the blood fails to clot. Now you've done this last week. Remember, recessive allele is found only on the X chromosome of the sex chromosome, not on the Y. Males only have one X chromosome. Remember again, just summarize it again. You can see there's a male, and males always are, here's a male, right? Okay, sorry, this is a female with the plus sign. It's a female, it's XX, and you can see two big chromosomes. This is the male with an arrow. It's XY. You can see the X is quite big. There's the XX and here's the X. And you can see the Y, how small it is. So always the disorders occur on your X chromosome of your gonosomes. Now they can show a karyotype chart and they'll say, identify the gender of this person. If you see two chromosomes of the same size and they're quite big, you'd say that's a female, because the X chromosomes are of the same size, and it can look like that. Then here you'd say this is a male, because you have one large and one small chromosome, and it would look like this. It can also be like a chromosome like that, but it's very small, so that will represent a Y, and that is a male. And remember, we said that for this person now, he's a male, but he got one X chromosome from the mother and one from the father. You see? So that's why she's a female. Here, this individual, he got one X from the mother, and obviously he got the Y from the father. So that is why now this tells you what? The gene for gender. One from the mother, one from the father. So this becomes an allele. That's one allele and that's one allele. That's the X allele, X allele, X allele, Y allele. Okay? Right. So let's move on. Now we're going to look at what are the sex-linked traits. Traits controlled by genes on the sex chromosome, which is the gonosome. On where will we see it? On the 23rd pair. 22 pairs are autosomes, but the 23rd pair will be your gonosome. Always learn that biological term. Most of the traits that are sex-linked or X-linked, the Y chromosome is tiny, so there's not many genes on it. So you don't really find any, you can't say Y to the power something. It's always X, because of the X chromosome. The symbol for the allele is attached to an X or Y, but you don't see anything on the Y because it's too small. Here you can see a genetic cross now. Remember, you've done this last week. You can see Y. Notice there are no alleles on the Y chromosome. Here you can see X. You can see that this person is a male, and you can see his X to the capital H. So we needed to see in the question paper, if X to the capital H is normal, then that's a normal male. And then here is a female, XX. She's a female, and here in the female, you can see she's a normal female, but she's carrying the hemophilia allele. So she's a carrier of hemophilia. So in a phenotype, she looks normal, but she's carrying the possibility of her children becoming hemophilic, because on her other X chromosome, you can see the recessive hemophilic gene, but the normal allele is dominating that recessive allele. So when you do a genetic cross, you remember, you put it in a Punnett square, which is far more easy. You draw actually 3 rows and 3 columns. In your top left-hand column, you write gametes. You put 1 allele per block. So you put that allele in this block. You put that Y allele into that block. You take this allele now from the mother and you put it in this block under gametes here. And then you put the other allele here. You see? Like that. So that's big H and small h. And then you say, the first to the first will give you this XX. The first to the second will give you that. So this in line will give you that. That in line will give you this. Then what happens if this allele of this person crosses over with that allele of the father? Then you're going to get like this, okay? And that going to give you this. So that's how you work a genetic cross. So this is basically showing you that sex-linked diseases are always represented by an X chromosome. And we talk about males and females in a sex-linked disorder, and there'll never be an allele on the Y chromosome. Right. So now we move on. This is because males only have one X chromosome. There's no allele to dominate over the recessive allele. So therefore, males are more affected by a recessive sex-linked disorder. So if a male is like this, see, he's carrying a recessive allele, which means now he's going to be having that disorder. Okay, it can also be like this. It can also be like this. It means that this person now is carrying that disorder. Or if he's like this, right, and capital H may mean that disorder is caused by a dominant allele. Then this male, if this X to the power H means it's caused by a dominant, it's causing a disorder, then this to the X to the power H means this male has the disorder, but it's caused by a dominant allele. This means that this male has a disorder that is caused by a recessive allele. Can you see that? And that means now this, if this capital X to the power H means it's a disorder caused by a dominant allele, that means this is a normal male. Because that means small letter recessive is normal. So you must read the questions very, very well. Ask yourself, this is a sex-linked disease because they got X to the power something. Right. They tell you that this disorder is caused by a dominant or a recessive allele. So the disorder, if it's dominant, then all of them will be represented by X to the power big H. If the disorder is caused by a recessive allele, it's going to be X to the power small h. So here you can see this is a female. Why? We got two X chromosomes, right? Then if this is X to the power D and X to the power D means normal, it means she's a normal female, but she's carrying X to the power small d. Depends on the question, what they'll tell you. Is X to the power small d colorblind? That means she's normal, but she's carrying the recessive allele for color blindness. This one here is a male, and if it's an X to the power D is normal, it means he's a normal male. Here you can have the other possibility. She's a female, it's XX, and if X to the power D is color blindness, it means now she's completely color blind, because on both her X chromosomes she's having the color blind allele. So she can give the color blind allele to her daughter, and she can give it to her son. Here it means the second one here, this male, look, this male here, he was normal. But when he has X to the power small d and X to the power small d means colorblind, it means that this male is colorblind. And here he is normal. This female is hemophilic because she got two recessive alleles on both her X chromosomes. And this woman here, she's normal because she got only the recessive allele on one X chromosome, and the other X chromosome is actually masking. The dominant allele of normal is masking the recessive allele. Okay, so that's how we represent our alleles. And by writing these alphabets with the powers, it means that this all represents what? The genotype, the possible genotypes of an organism which has sex-linked characteristics. Right. This is what happens when you are colorblind. You are unable to differentiate between like red and orange, yellow and green. Okay, certain colors that look similar, you won't be able to see it because there's a disorder in the retina of your eye, which you're going to learn later in the year. So you can have a red-green color deficiency. You can be hemophilic, which is X-linked. See, color blindness is sex-linked or X-linked. It's inability for blood to clot in hemophilia. Then you have the Duchenne muscular dystrophy, where your muscles begin to weaken from the age of at least 3 years, right? All the muscles of the heart and all the any muscle in your body begins to weaken because of this genetic disorder here. And it's actually caused by a recessive allele on the X chromosome, right? Fragile X syndrome, these are other sex-linked diseases, and that Fragile X syndrome is actually caused by a dominant allele, and it results in mental disorders. All right, it affects the brain. Then Rett syndrome is caused by a dominant allele. So let's say if I put Rett syndrome, and Rett syndrome is actually caused by a dominant sex-linked disease. Then I must put X, and maybe I can put capital R because it's caused by a dominant allele. That means X to the power small r, which means this person is normal and this person here has Rett syndrome. Let's look at fragile X syndrome. So let's put it as X. It's a sex-linked disease because you can see it's X-linked and it's caused by dominant allele. So let's put capital F. It means this person has fragile X syndrome because it's caused by a dominant allele. Let's look at Duchenne muscular dystrophy. Then it's sex-linked. They're telling you in the paper it's sex-linked, but it's recessive. So let's say you can put by Duchenne muscular dystrophy, I can put X to the power small d. It means it's caused by a recessive allele. And hemophilia, I can put X to the power small h because it's recessive. They will give you this alphabet. You don't use your own alphabets, okay? And color blindness, I can put it as maybe X to the power small c, right? But they won't use C because a small c and a big C, you can't really differentiate. So they'll give you their own words. For example, they can put X to the power, let's say the power small t. And they say that is called by, that's color blindness, X to the power small t. Here's color blindness, okay? X to the power small t. That means X to the power big T means the person is normal. All right, so you know how to represent your alphabets. That's very important. Now we're going to look at the inheritance of hemophilia. The father can only pass his affected alleles to his daughters, right? And then the mother can pass her affected alleles to her sons and daughters, and we've seen it in the previous slide. You can see there's the father, there's the mother, and then you can see their father is always XY. We're dealing with sex chromosomes, XY. Mother is XX as a sex chromosomes. Then how will it happen that if a mother who is a carrier, can you see they put different colors because this man is normal without hemophilia, so he's very normal, it's full gray. But this one, she's gray and red, means she's normal but she's carrying the hemophilia allele. All right, so how will their children genotypes be? So remember, if there's a son, so this father takes his Y and gives it to the son. Here's it. Then the father can't give his X and his Y to the son. One must come from the mother, one must come from the father. So he's already donated his Y, which comes from the sperm cell, to this offspring. Then obviously the other chromosome must come from the mother, and she only has X. So she can either give that X there, or she could also give this X here. And here she gave it to the son here. Can you see that? Now, if she gives the one with hemophilia, that means that this son is going to be hemophilic because he's carrying the X-linked X chromosome there. And you can see this is the daughter. The daughter was going to get one from the father. Can you see? There's his normal, here's his X chromosome. Or she can pass, the mother can pass the other X chromosome to the daughter. Or she can even pass it like that. Can you see that? So mothers can pass the X chromosome to either the daughters or the sons. Can you see? She's the one that's given it. Here she's given her normal allele to this son, and here she's given that normal allele to that son. So it depends how many children she has. She can have 40 children. It doesn't mean that you can only have 4 children. It means you can have, no matter how many children, you can have 15 children. But this is the only possible genotypes of the children. That means that they can go to a counselor and say, we want to, what is the possibility of our children having hemophilia? We both are normal. My husband and my wife, the husband and wife are both normal, but you know, we tested our blood and we've seen that the mother has a hemophilia allele. We want to know what is the percentage chance of any of our children, whether it's a boy or a girl, what's the percentage chance of them being hemophilia? So the genetic counselor will work out a cross for the parents and say, fine, you are completely normal. We did a blood test. The mother is normal, but she's carrying the hemophilia allele in her blood. So she's a carrier. So your possibilities are that you can have a 50% chance of having daughters, always. 50% of having daughters, and there's a 50% chance of having sons. But out of the 50%, you can see that out of the 50% daughters, see, 2 out of the 4. that means 50%. 2 out of the 4, or 50% of the daughters. 25%, see, you're going to have, okay, the daughter, we're not looking at children, right? So out of all the children, there's going to be a 50% chance of the daughter and the sons having hemophilia, of the children. When you talk of children, you talk of sons and daughters. But if we want to know, if we're going to have just daughters, so we can ask a question, what percentage of the daughters are having hemophilia in this chart? It's going to be this one here, she's going to have the hemophilia allele, and this is the normal allele, which is dominant. Ne? So this female, she's going to be just like her mother. She's going to be normal, but she's carrying hemophilia allele. That means she's going to be normal. That's the phenotype. Let's look at the son. Here the son is going to be also normal because it's gray. He's got the dominant allele, which is normal. Right, this daughter, she's getting the normal allele from the father, and she's getting the normal allele, the other normal allele from the mother. So she's also going to be normal. But this son, the mother is carrying the hemophilia allele, but this son is getting the Y chromosome from the father. But now he's getting the X chromosome with the hemophilia allele from the mother. So that means he's going to be looking like this. So he is going to definitely have hemophilia. So that means out of the 4, 1%, 1 out of the 4 will have hemophilia, and that one is going to be a son. So no matter how many children they have, if you have a son, there can be a 25% chance of your sons having hemophilia. Ne? But when it comes to your daughters, no matter how many daughters you have, none of them will have hemophilia. So your daughters will be normal, but there is out of the 100% daughters, 50%. That means 1 out of the 2 daughters, 50% chance of the daughters can carry the hemophilia gene. And you understand, children? So 100% means the 2 daughters, but 50% can be a carrier, and the other 50% can be absolutely homozygous normal. So you read this as a hemophilic son because he has X to the power small d or small h. That's the hemophilia. It's showing in his phenotype because his Y chromosome cannot mask it because there's no alleles on the Y chromosome. So he's going to be hemophilic. She's going to be normal because she's carrying the normal allele, which is masking the hemophilic allele. She's going to be normal. He's going to be normal. He's got the normal allele from the mother here, and the allele from the Y allele from the father. And this daughter here is going to be absolutely normal, homozygous normal. So that is this normal hemophilia is description. That's the phenotype. Writing these alleles are actually the genotype. All right. Now let's look at it. This is a baseline assessment where now you've done sex-linked diseases. So let's look at it. Duchenne's muscular dystrophy is a genetic disorder in which the skeletal muscles progressively weaken. It is a sex-linked disorder. That means it's got already, we know it must be having an X, right? And because it's a dystrophy caused by a recessive allele, and let's say now here in the question they don't give it to you, but let's say I'm going to say that Duchenne's muscular disease is caused by a small letter. So it's going to be X to the power small letter, not just D. Not just D like that. Okay, but it's sex-linked. And they tell you it's a sex-linked disorder. That's why we must put X. Duchenne's muscular dystrophy mostly affects boys and causes their muscles to weaken from the age of about 3. By the time they are teenagers, they'll use a wheelchair, and eventually the heart and respiratory muscles, that means the breathing muscles, will be affected. Learners, you must learn to read the entire paragraph well, because in the exam, they're going to ask questions based on the paragraph. All right, so you must read it very well. Don't just browse through it. Read it very well. Define a sex-linked disorder. What is it? It is when there is a genetic mutation caused on the X chromosome of the sex chromosome. So what's a sex-linked disorder? It is when you have a gene mutation on the X chromosomes of your gonosomes, then it's going to become a sex-linked disorder. Right. According to the extract, when will parents first notice the changes in the muscles of the child? And you can see around 3 years of age. All right. Explain why it is more common for boys to suffer from Duchenne's muscular dystrophy than girls. Why? Because remember, if you're a boy, you are XY, right? Now, obviously, because it's caused by now, if you're a son, you're going to have the Y from the father, okay? And then obviously, if you're going to be a boy, you're going to have the smaller, you're going to get it from the mother. So a mother has to carry this Duchenne's recessive allele. So when she gives it to the son, he's going to get that. That comes from the mother, okay? And because men or boys have just one X chromosome, they do not have another X chromosome with a dominant allele to mask this disorder. That's why if they get one X chromosome from the mother and she's affected, he will also be affected because there isn't another chromosome to mask it. Like if it's a female, it's like this, then can you see the dominant one will mask? You'll see the dominant normal. You won't see this Duchenne's disorder in a female. But in a male, you'll see it because there isn't another X chromosome. Right, so let's look at the answers to the same questions. Define a sex-linked disorder. It's an abnormal condition or it's a disease determined by an allele on the gonosomes or on the sex chromosome. Basically, you have to say it is it's a gene mutation that will cause a disease, and due to this gene mutation on the X chromosome, it's called a sex-linked disorder. Right, and then we're going to see the next question. When will the parents first notice it? At the age of 3, right? Explain why it's more common for boys to suffer from Duchenne's muscular dystrophy. Because you must say that this disease is actually a gene mutation on the X chromosome. Boys only have one X chromosome. So if that chromosome carries the Duchenne's gene, you won't say that, not gene, it's allele. So if that X chromosome carries the Duchenne allele, the boy will have the Duchenne's muscular dystrophy because there's no other allele to mask it. Okay? So remember, boy, you're going to say Duchenne's disorder is caused by a recessive allele on the X chromosome. Males have one X chromosome. Therefore, if the X chromosome has that allele for the disorder, it will show in the phenotype. The terminology that we've done, and my colleague has actually explained earlier, we know codominance. Now we're going to see it in blood groups, whereby you're going to see how 2 alleles will be represented in your blood, and that's codominance. For example, blood group AB, you're going to see that in your blood group, this person cannot just be A or B, but it's going to be AB. You know, that's a gene. A gene is actually an inherited characteristic that you get from your parents, right, caused by the dominant allele. And gene mutation is a sudden change in the sequence of a nitrogen base of a DNA. So remember, children, there's a very big difference between gene mutation and mutation. A mutation is when there's a sudden change in the genome, the genetic makeup of a person. That's anyway. But a gene mutation is when there's a sudden change in the sequencing, the arrangement, the sequencing of a nitrogen base of a gene of a DNA. So there we're talking about where AGA now changes to ATA, where the G guanine now was replaced by thymine. So that sudden change in the sequencing of the nitrogen base in the gene of a DNA, that's called a gene mutation. And that sudden change from AGA to ATA will actually change your amino acids, change the protein, causing a disorder. Right, that's very important. Now let's look at the 3 rules of blood groups. 1 to 3 rules of blood groups. First of all, learners, we can see that every individual has only one blood group. An individual has 2 alleles for that blood group. So you must have you must have 2 alleles for any gene. The gene here is blood group. There are 3 different alleles controlling blood group. So normally we said there's 2 alleles that control a gene, but in blood group it's different. There are 3 alleles that control the gene called blood group. And because there's more than 2 alleles, we say that blood groups is an example of multiple alleles. There are 4 blood groups. So we have 4 blood groups, but controlled by 3 alleles. So since the alleles are more than 2, there are more than 2. We say blood groups is an example of multiple alleles. We're going to see the blood groups now. So, the inheritance of blood groups is an example of multiple alleles. Why? More than 2 alleles control blood groups. The notation for alleles indicating blood groups is only I to the power A or I to the power big B and small letter I. So where we said sex-linked diseases was represented by X to the power something. Now in blood groups, we use the letter I. Okay, because of your kind of a protein found in your blood that starts with I, immunoglobulins, right? Don't worry about too much of those things, about that name, but you use capital I or small I for blood groups. That's all you need to know. Right, now we're going to see the different combinations of the alleles resulting in 4 blood groups. So you can have 4 blood groups in a population. And here's your blood group. So when you say blood group A, blood group B, blood group AB, blood group O, these blood groups are phenotypes. You're writing it blood group A. But to represent the phenotypes, we must use alleles. We must use alphabets. So can you see, when we use alphabets, it's the genotype. So this means here, what does it mean? It means capital I to the power big A. Capital I to the power big A. What it means is this person is blood group A. Blood group A. But for him to be blood group A, he got 1A allele from the mother and 1A allele from the father. What else does it tell you? It tells you that capital A means blood group A like that. It's not because they wanted to write it, it's like that. Capital A can never be written as small a. That's wrong. A blood group A is a dominant group, and because it's dominant, it's always going to be represented by a capital A. But to represent blood groups, we must use a capital I. So capital I is dominant. This means this person has a blood group and he is blood group A. But his blood group A, his mother was blood group A, his father was blood group A, and he is blood group A. So that means he got 1A dominant allele from the mother or 1 dominant blood allele A from the father. See? So 1 dominant allele from one parent, another dominant allele from that parent. And because he's blood group A, we say both of the parents have the same blood group. So he has the same blood group as his parents. That means he is homozygous for blood group A. Okay, then this person is blood group B, and we're going to come back to this blood group here just now. This means this person's parents were both blood group B. So this allele came from the one parent, that one came from the other parent. So he is homozygous blood group B. Right, we're coming back to this other one just now. Let's look at blood group AB. Blood group AB means A and B are dominant alleles in blood group. It means one parent got an allele, an A allele. So one of his parents, either the mother or the father, was blood group A. So she donated her A allele to the son or daughter, whoever this person is. And the other parent was blood group B, only blood group B, and therefore he donated his blood group B allele to this. So this person now, you can see is AB. And AB is co-dominant because you can see 2 alleles expressed in the blood. So he's co-dominant. And the other blood group, which is a very important blood group, this is the person that can save lives, is a person with blood group O. And that means blood group O is a recessive. It's a recessive blood group, and it's represented by 2 small i's. So recessive blood group is 2 small i's. Okay. Now what does it mean by this? Here we mean now, if we're looking at this here, this means that this person is heterozygous blood group A. Heterozygous blood group A means one parent was blood group A and the other parent was blood group O. Can you see, to represent blood group O, we use 2 small i's. So learners, what are the 4 blood groups? Blood group A, B, AB, and O. Out of the 4 blood groups, which blood group is recessive? It's I. which means that this person can donate blood to anybody. See, he can donate blood to somebody with blood group A, and that person will still be blood group A, because the I, which is represented by a recessive blood group, it's recessive, so it's actually not going to affect the blood group A, because A is dominant. And blood group O can also donate blood to a person who's blood group B. Can you see that? Because if he donates blood to blood group B, then he's donating blood, but the person is still, his phenotype is still blood group B. But O cannot donate blood to a person with blood group AB. AB must only get blood from blood group AB, not from A and not from B only, because then the blood is going to stick. So this O can only give blood to a person who's blood group A, or a person who's blood group B, but he cannot, and also to a person who's blood group O, but he cannot give blood to a person with blood group AB, because nowhere in a person's blood group AB you can see the recessive allele for the blood group. Right, and there you can see that's complete dominance, and that's complete. So here you see co-dominance, complete dominance, blood group A dominates, blood group B dominates, blood group AB dominates, both are seen together, so it's co, and here it's complete dominance, completely O, because both the recessive alleles are O alleles, and it's showing as blood group O. Right, and then let's look at this question quickly. A man with blood group AB and a woman who's heterozygous for blood group B plan to have a children. How many alleles control the inheritance of blood group? It's 3 alleles. What is it? It's allele A, it's allele B, and it's allele O. We represent allele O with a small letter I. That's 3 alleles, okay? Describe the type of dominance that occurs in the inheritance of blood group B in a woman. What's the type of inheritance? It's going to be complete inheritance, complete dominance, okay? So you can see that how many alleles control the blood group? It's 3. Describe the type of dominance occurs in the inheritance of group B. It's complete dominance, because if she's going to get B from the mother and B from the father, she's going to be blood group homozygous B. If she's going to get B from the mother and the small I or small letter or the blood group O from the father, it's recessive, she'll still be blood group B. So complete dominance for blood group B, because O is recessive. Now this is almost the last question, children. Mr. and Mrs. Fonella are concerned that the baby girl does not appear to resemble any of them. So they went to the hospital. Mr. Fonella is blood group AB, Mrs. Fonella is blood group B, and the baby that we given is blood group O. So for Mrs. Fonella, right, her blood group is B. She can be like this, I to the power big B, I to the power big B, or she can be I to the power big B, can you see? And she can carry the small recessive allele. The baby girl can only be 2 small letter I's. So now we know already, you must work out the genotypes already of the parents before you go to the questions. We already seen Mrs. Fonella is blood group B. Okay, she can only be homozygous blood group B or heterozygous blood group B. For heterozygous, she must carry the dominant allele for B, and she must carry the recessive O allele represented by small I. Baby girl is O, and then to represent the genotype, it's going to be 2 small i's, because blood group O is represented by small i's. Now explain why the baby girl with blood group O cannot be Mr. and Mrs. Fonella's daughter. So let's look at Mr. Fonella's blood group. He's AB. Mr. Fonella is AB. That means he's I to the power A and I to the power B. Big capital B. So if this baby is this small I, that means Mrs. Fonella's blood group, for her to be B, she had to be this genotype. She couldn't be this. So if this was, she could be this or she could be this. Now, let's say if she was blood group B, heterozygous, then she could have donated this I allele to this child. But then the father had to be having this small I as well. But he doesn't. So this can never be the baby, because the father, Mr. Fonella, does not carry the O recessive allele represented by small I. Mr. Fonella is co-dominant. He does not have the recessive allele. So this can never be the baby, their baby girl, because the mother could have donated the O allele, but well, where's the other one from the father? It's not there. Okay, so let's look at it here. Give the possible genotypes of Mrs. Fonella. We said homozygous B, or heterozygous B. Let's look at the baby girl's phenotype. We said she's homozygous blood group O, represented by 2 small eyes. Then why is she cannot be their daughter? Because the baby inherits the allele from the mother and the father. Né? So if she's blood group O, she can inherit the O allele from the mother, but Mr. Fonella does not have the O allele. So therefore he can never be the father. So explain why can't we use paternity testing to say, no, okay, we must only use blood group to determine who's the father of the child. Many fathers could be blood group I to the power AB. Many fathers could be blood group B. Many fathers could be blood group A. But now, what did we use blood group for? We can eliminate. We can say, no. This child can never be the father, the daughter of this, because his blood group is AB. Therefore, the child doesn't have any of the alleles of the father. So you see, we can use it to eliminate the possibility of that sibling not belonging to the parent or that parent not belonging to the child by looking at their phenotype. So remember that we can't use blood groups because blood groups are used to exclude a certain individual. But it cannot confirm that that person is the father because a large population of people can have the same blood group. Use your knowledge of sex chromosome and explain why the sex of a child is determined by the male gamete. Why must be? Because any individual, whether you're a boy or a girl, you must have an X chromosome. So that can come either from the mother or the father. But for you to be a boy, it has to come from the father. So therefore, a father is a male. Therefore, the sex of the child is determined by the male gamete. But if you want a boy, then obviously the male has to have the child has to be XY, and that Y has to come from the father, and that Y comes from the sperm cell. Remember, the sperm cell must have the last chromosome as being Y. Remember, children, you did somatic cells, you have 2 chromosomes, but a gamete must have half. So the sperm cells can either be 22 autosomes plus 1 gonosome, which is X, or 22 autosomes plus 1 gonosome, which is Y, in a sperm cell. Né? So the sperm cell with the Y must actually fertilize with the sperm cell with the ovum with the X, and X is always an ovum. So here you're going to get 22 plus 22, 44, and X and Y, and that gives you a boy child. So it is the sperm cell that determines the sex of the child. If you want a son, it means that the sperm cell is not, that means all the children that are forming, it's coming from the sperm cell with the X. So there's something wrong with the sperm cells. This parent has more sperm cells with 22 plus X and very few sperm cells with 22 plus Y. So it is the male gamete that determines the sex of the child, okay? Normal females have 2 X chromosomes. Normal males have an X and Y. The female always provides the X in the ovum. If an ovum is fertilized by an X-bearing sperm like that, then it's a female child. If an ovum is having a Y chromosome, then it's a male child. Okay, and that actually tells us, now we spoke about this children color blindness. We've done that is caused by a recessive allele on the X chromosome, right? And you can see here, when you do a genetic cross, it's P1 phenotype, you describe genotype, you use alleles, you have meiosis, then you describe your gametes, then you show fertilization in a Punnett square. You always put 1 allele per block. You always write gametes there. You can see if he's a colorblind man, it's XY because he's a man, but he's colorblind, recessive allele, so his X chromosome must have the small letter. She's normal female, she's homozygous, then she's going to have 2 capital letters. She's for normal, X to the power big B, and you put it like this. But the male, you can put 1 allele per block. Either on your left, you put it on the 1 per block, that there, this one there, and then the female, you can put 1 allele there and 1 allele there, and then you cross it, okay? So this is how you do a normal genetic cross. Then you write down the F1 means the offsprings, and you describe the offsprings. That this is the genotypes, you get a mark for it, okay? That there's 1 female, she's going to be normal. That's a phenotype. You cannot say she's normal but a carrier, because you cannot see a female carrying the allele. So you look at the phenotype, normal female, normal female, normal male, normal male. So in this kind, where you have a colorblind man and an absolutely homozygous normal female, all the children will be normal, But now you can see in this one here, this genotype, when you test the blood, you can see this female is carrying the color-blind allele. She's carrying the color-blind allele. But the male will, when he donates his X chromosome to the daughters will never be colorblind because the X chromosome from the father is normal. All right. And here you got 2 normal females and 2 normal males. Can you see they're not saying they're carriers because you're looking at the female's phenotype. Right. P1 and F1, you know you get for P1, and writing F1. This genotype, you get a mark, right, for P1 and F1, meiosis and fertilization. Remember, meiosis must come before the word gametes. You can't get gametes and then and then the meiosis occurring. So the steps have to be ordered like a recipe. P1, the phenotype, then genotype, then the meiosis, then the gametes, then the fertilization represented by a Punnett square, then the genotypes of those offspring, which is F1, then describe the genotype, which is your phenotype. That's it, right? And here's your homework, learners. And this homework is a very simple homework. You can get it from your worksheet, and also the answers will be provided to you. But just look at it. Let's just look at it. It's an investigation to determine which blood group is dominant in a community. Okay, so they collected information about the blood groups of 200 donors in each of the 3 blood groups, 3 blood donor clinics. Okay, so they collected information about blood groups of 200 blood donors in each of the 3 blood donor clinics in the community. So there are 3 blood donor clinics, and in each clinic they used 200 donors. So altogether the sample size was 600. Now that's quite a large number compared to 5 or 10. So by having a large sample size, what did the investigation cater for? Reliability. Right. They did not include first-time donors in the investigation. Why won't you go and include a donor who's coming to donate blood for the first time? Because the blood still needs to be tested. What if the blood group isn't yet known. The blood group is not yet on, they're not yet registered on the database. So you can't have the first group of blood donors to be part of your investigation because you haven't established those donors, you haven't established authenticity of the blood or what the blood group is or whether they're having certain diseases, and they're not part of the database. So you will exclude those first-time donors for those reasons. You can see this is a pie chart. It has sectors, it has labels, it also has a heading, percentage of each blood group in the sample. And remember, what's the total sample now? Not 200, because you remember they took 200 for different clinics per clinic. So altogether it was 600. Right. Now here you have 200 blood donors in each of the 3 blood donor clinics. Don't think it is 200 donors of each blood group. Né? It is 200 donors per clinic. So now you're going to have blood group O. You can see it's 55%, blood group A 25, and you can see the smallest blood group there was blood group AB. And now let's look at the questions that follow. State the aim of the investigation is going to determine the blood groups in the community. What are the planning steps? Whenever you do a planning step, children, whether it's plants or whether it's animals, this will always be there. Remember, you cannot ask for permission from plants. So when people are involved, you're going to say you're going to first get permission to do an investigation. Then you're going to determine when the time and the date of the investigation. When you're going to do it, what date, what day and time. Then you're going to determine how you're going to collect your data and how you're going to record your data. So that's always there. The tools you're going to be used to collect your data, when you're going to be doing the investigation, and what your sample size will be. Okay, and remember, give one reason why they did not include the first-time donors, because the first-time donors are not yet authentic and they're not on the database. And here, learners, you can see, calculate the number of participants that had blood group B, show all workings. So you're going to actually look at the chart again, right? And then you're going to see 600, if it's 15%, you'll say 15%, 15 over 100 times 600, and that's going to give you the blood group in question. And remember, sex-linked diseases are sex-linked disorders. Genetic problems have sex-linked disorders are hemophilia and color blindness. blood grouping. Remember this, it's a multiple, caused by multiple alleles, and that we have blood group I to the power A and I to the power B are dominant, and small letter I is recessive. Okay, so the next lesson, learners, will be the just-in-time lesson on the 10th of April from 2 to 3:30, and then we're going to be discussing pedigree diagrams and genetic engineering. Thank you.